So I thought, well, what am I going to do? This month I was looking at, I got an Amazon Fire a couple years ago for $35. It's a very nice machine. Okay, nice tablet. And I said, oh, maybe I should learn how to program that and write an app for it or something. Didn't work out. I couldn't get the Angel thing working. So that was the end of it. And then I said, okay, I got the E4 running, E4 compiled on C and running on Visual Studio. But I was only able to get it onto the Visual Studio as a console application. Okay, so you just give a very generic the MS-DOS type of screen and have the force running on it. So that's too primitive. So I thought, well, can I get it to run a window, run a better window? The problem with the console is you cannot size it. So when the dump is too big, it just falls over. If I have a better console, I can scroll much more information on it, just like what the Arduino gave me. Such as, let me try to do the kind of Arduino serial monitor type of display. It didn't work out. Okay. So the Visual Studio has what they call the window form application, and you can open up a window real easy. Okay, but then the Windows form is based on C#, and so I got a window there, but I couldn't convert the C code or C++ code into the C#. And then I started look for why can't I do this? Why can't I do that? And one guy said, "Hey, guy, C# is not C++. Okay, there are different languages." Okay, so that didn't work out. So I thought, well, Win32 Force use the Win32 interface, so it opens a window pretty nicely. But then I look at Win32 stuff. It's just too complicated. So finally, I decided I can't do Windows. Okay, so I go back to the Genome, and so that's my failure in 2016, and so what can I do now better than what I've done in 2018? So and I think the problem I had in 2018 is that I was kind of focused on the microRNA. Okay, so we got lots of microRNA in many different species, and so I was trying to use the collection of microRNA and try to search the microRNA information in genomes of bacteria and simple living stuff. But the database of microRNA has 57,000 microRNA from 271 different species. I thought that I should probably share different species would share the same set of microRNA. But it turns out that they share a lot, but not enough. And 57,000 species microRNA is not enough to cover all everything. So if you just having a very small set of microRNA and you search the big genome and you don't find much matches, and that was the problem. So I thought, well, I was search blindly in the genome of the very different species, and I think that's probably not a good way to do it. So this year, I would focus on long non-coding RNA, and these are uh, uh, expressed genes. These are genes that actually are found in living tissue, and they are expressed information. So these information are things that's being used by the living cell, and they would contain useful information rather than the genome, which contain lots of junk. So I will talk a little bit about the bioinformatics on DNA, RNA, and so I was searching for pairs, uh, 20 base pair patterns. And it's like microRNA, but if I open up my search, not just limit myself to existing microRNA, I may be able to find um, many more matches and find necklaces, lots of pairs should be uh, should be uh, placed next to each other into necklaces, and these are probably uh, the uh, the basic mechanism the cell would operate. Okay, so this is I'll show you what I've found. So here in 2008, I hope to find microRNAs in the DNA genomes of simple cells like bacteria and nematode. So repeated pattern of 20 base pair found in DNA genomes did not match known microRNA. I found a few matches, but not good enough to support my model of the cell computer. So, this month I'm focused on the long non-coding RNA, and these contain valid information that cell uses for its operation, and these are expressed. So all the junk stuff has been deleted, and so in the long non-coding RNA, we got much more useful information in it. So I may have to focusing on that would probably give me much better results. So the DNA, we have genomes. Every cell has a genome. It's a DNA sequence contained in chromosomes. Okay, the higher living creatures has more chromosomes, and the bacteria and viruses they only have one. And the coding DNA in the human being, at least in our DNA has been studied most exhaustively, and in the human DNA, only 2%, less than 2% of genome are expressed as messenger RNA to produce proteins, and the rest of it, greater than 98% of the genome, is non-coding DNA. So, well, it used to call junk DNA. They have no function. But now it has been changed, and people have thought that, well, there are useful information in them, but we just don't know. So the RNA is what's been transcribed from DNA into RNA molecules, which are used in the cell operation. Okay, so there's many different RNAs, and the most important type is messenger RNA. And these RNA, there are thousands of base pairs, and they encode proteins. Okay, so that we know, and that's the coding RNA. And beside that, we have a transfer RNA and ribosomal RNA. And we know these RNAs are basically part of the ribosome system, which builds a protein from the messenger RNA. So these are all the RNAs that's well understood. But the rest of it, there are long pieces, there are short pieces of RNA, which are not well understood. Okay, so the long non-coding RNA, and they are just arbitrarily classified as 200 base pair or more, and they could be very long, thousands of base pairs. And there are also very short RNA, the microRNA, which has 18 to 22 base pairs. And originally they said, oh, these are just junk stuff that's left over when we process when you break up a cell, and you probably break up lots of things. And that when you break up the RNA, you got the little pieces of microRNA. But later on, they found that these microRNA are not just random scraps, and many of them has very important biological functions. So microRNA has been studied quite extensively, and lots of Nobel Prizes were awarded for the work on the microRNA. And besides microRNA, there are lots of other small RNAs which has different functions. Okay, but our understanding of the long non-coding RNA is just completely blank. Okay, but people know there's lots of them. Let me see. Let me just go follow the slide. Okay, so basically, what happened is all the information are on the DNA, on the genome. And the useful information are transcribed from the DNA to RNA. And then RNA carrying its information would perform various functions in a cell. So the messenger RNA produces protein, transfer RNA, and ribosomal RNA, they assist protein production. And the microRNA, at least we have found many microRNA which does control the expression of messenger RNA. It will enhance or impede the messenger RNA expression. And we got lots of long non-coding RNAs, and nobody knows what they are doing. So my proposition of a cell computer is that each cell I maintain is a complete computer. Okay, the computer has code, so the code is stored in DNA. And what kind of form of this code? And I would propose that it's like this cell computer uses instructions like microRNA. These instructions are, 18 to 24 base pair long, and that's long enough to code lots and lots of different uniquely code many different microRNAs, and these many different functions would go out into the cell and cause various things to happen. Okay, so the basic instructions are in the microRNA, but microRNA are formed through long non-coding RNA. So long non-coding RNA contains lists of microRNA. Okay, and some microRNA we knew that they transcribe into messenger RNA and they produce protein. And these are the primitive instructions in forth. And some microRNA transcribes long non-coding RNA. And this long non-coding RNA contains a list of microRNA, and each of them would perform their own work. So for complicated functions, you need long linear RNA, which contains many microRNAs, and each microRNA may call upon another long non-coding RNA to perform even more complicated thing. Okay, so this is just like a fourth. We are doing nested list processing. Okay, list call, list call, list call, list. At the end of the list, you call a primitive instruction to do real things. Okay. So to prove the existence of a cell computer, I have to demonstrate that the long non-coding RNA contains lists of microRNA. Okay. And my failure last year was that I was focused on the existing microRNA, and that's not enough. And known microRNA are not enough to prove the above hypothesis. So I need to extend this space. I would go over and above the known microRNA and look for pairs. Look for 20 base pair patterns, which might be microRNA. So the collection of long linear RNA are complete enough to prove my thesis, because in human genome, people have identified lots of non-long coding RNA, and these are actual RNA found mostly in brain. Okay, so if the collection of long non-coding RNA almost cover the whole genome. Okay, so if we do exhaustive search of these 20 base pair patterns in all linear RNAs, we might yield a collection, a more or less complete collection of pairs, or the potential microRNA. And I need to show that the linear non-coding RNA contains lists of the pairs. And so that's so this is what I proceed to do this month. Okay, so there's what is information or bioinformation? Information is any information, okay, in any form. They're just repeat patterns, okay. And if you find random patterns, there's probably nothing there. But if you find repeated patterns, there must be something there, okay. So the repeated pattern contains information. So in the genome, in the coding DNA or coding RNA, we knew that 3 base pair code. There are lots of three base pair code which are code for amino acids. And we also know that lots of these three base pair code are form consecutive 3 base pair code information to build proteins. So these are the coding DNA we understand. But outside of the protein, we knew very little. So I assume that if there are information in the junk DNA, in the long non-coding DNA, in the long non-coding RNA, these are possible, very likely possible patterns that form the basis of a computer. So I call them pairs. So long non-coding RNA. In the people studied different cells. They just break up the cell, and they can extract lots of RNA from the tissues. And 80% of the RNA they found in the laboratory are non-coding RNAs. Okay, only less than 20% are coding or messenger RNAs. And interesting enough that the most abundant RNA, this long non-coding RNA, are most abundant in testes and in neural tissues. Yeah. That's why most of the long non-coding RNA are found, are extracted from brain. Okay, and so 80% of the long non-coding RNA are tissue specific. Okay, so they use different tissues and found different collections of long non-coding RNA. So from there we know that the long non-coding RNA contains important information for different cells to operate differently. Okay, we don't know what they are doing, but we know that they are doing something important because we found them different long non-coding RNA in different tissues. For in the human genome, they have spent lots of time analyzing the DNA and associated the RNA information. And in one of the collections, they have identified 270 thousand different RNA transcriptions. And this is the database I kind of used because the collection are kind of there's a lot of different collections, and it's kind of hard to can't use them all, but this seems to be a very nice collection in Cambridge. So it has lots of different species, and this genome 38. This is the human genome. In the human genome, they have identified lots of non-coding RNA, and this file is 77 million bytes. And it has a much larger collection of cDNA, and this is almost 300 million base pairs. So the cDNA is kind of interesting because let me see. Let me don't go into the detail of that. It's a nice story, but let's skip it over. So the human genome, we got the 3 billion base pairs, and then less than 1%, less than 2% are the coding DNA, and then beside the coding DNA, there are 200 million transcripts being reported, and among this transcript, only 20,000 proteins, 20,000 genes are identified quite consistently. Okay, and people already thought that we got 100,000 genes, but so far the genes, the proteins, protein coding genes, we have only identified 20,000, and they have 57,000 non-coding RNA. But over the years, they have found 189,000 coding DNA. The coding DNA is derived from the non-coding RNA. So they use the reverse transcription. And mostly they analyze the brain tissue. They break up the brain tissue, extract all the RNA out of it, and use the reverse transcriptase. And they turn the RNA back into cDNA. Okay, and the cDNA is much more stable, and you can do much better analysis on it. So lots of people have identified lots of the cDNA. So I'm not sure about these different non-coding RNA or cDNA. I thought that they probably contain the same information, but I don't know. So might just well analyze them all. Okay. So for the flatworm nematode, the genome is much smaller. So it's 100,000 bases. The bases are separated in 7 chromosomes. And in this, it has been studied quite exhaustively. And they have identified lots of 3,000 non-coding RNA, and the data file is about 2 million bytes. So these are the human model and the nematode. These are good models to test my hypothesis. So I have these are some old slides. You might have seen them. So I would search for pairs, and the pairs are 20 base patterns, repeated 20 base patterns. Okay, and I try to identify all pairs that are unique and repeated. And once I got the pairs, I will look to see how the pairs are distributed in the genome and identify necklaces. Necklaces are just clusters of adjacent pairs. Okay. So the idea is that the pairs are related to microRNA, are related to the basic instruction set of the cell computer. Okay, and the necklaces are just lists of pairs, and the non-coding RNA might be just a list of microRNA. And this is basically my model of the cell computer. So to search, you see this is the information is huge, okay, and searching this huge database, you need good code, very fast code, and of course you need a good computer. So big long non-coding RNA, it's just like when I do the DNA search. DNA, these are just huge files when you get to high level of living creatures. So I would break up a large data file into 4,096 threads. Okay, so it's the ACGT. These are bases. I just take I say, well, I would just try to search patterns with a unique 6 beginning base pair. Okay, each I got the six different bases, and that's each base is worth 2 bits. Okay, so 6 bases is 12 bits, and 12 bits is encodes 496 different patterns. So each of these patterns, I will build a thread. I search the entire data file and identify 6 unique base, and then build a thread. Okay, and the thread just start with the next code, which has the same 6 base, and then sum. Okay, so if I just build a list of these points, which has the same 6 base pair code, and this I got a thread. And when I search through this thread, I will get all the patterns which begins with the same 6 base pair pattern. Okay, yes.
Say thread, are you saying like a thread in an operating system?
No. It's just a list, yeah, just a pointer. Okay, so I collect all the pointers which points to the 6 base pair patterns in the same file. And when I'm searching for the patterns which begin with this 6 base pair, I only have to look at this list. And this will reduce my searching. Using this thread, I would only search, 1/4,000th of the entire data file. So it will reduce the search time by a factor of 4,000. Okay. So this is what I'm doing, and so the search time is greatly reduced. And I do because I'm using I have 2 computers at home. Both are bought in year 2006, and they're running on Windows 7. So these are not the biggest and fastest computers. But when the search algorithm is optimized, even these very old computers can do a decent job. Okay, so the DNA file and the long non-coding RNA file are still big, and I was able to break them up in 4 million base pair chunks because I really cannot search the the file when it's much bigger than 4 million 4 million bytes. Okay, so I have when I have a file which is bigger than 4 million pairs, I just search 4 million pairs in chunks and then combine the result afterwards. So the pairs in each chunk are identified. All unique pairs are combined, and then each from all the chunks, I would identify unique pairs, and then all the pairs in different chunks, then combine into the big pair database. And each pair would be assigned an ID. And this, once I have all the pairs, then I would search the entire data file again and see and locate all the pairs, and then look at this file with all the pairs identified, looking up for necklaces. Okay. So when you do this search for 20-bit unique and repeated patterns, you get a huge number of repeats. Okay, and there are very interestingly, there are the most abundant repeats are spaced by one base. Okay, so they are right next to it. Okay, so it's like you have the match, and it just match one after the other. These are basically caused by long stretches of repeated pattern. Okay, and in the when I'm doing the search for the genome for the DNA, I knew this will happen because DNA, there are lots of protein code, lots of messenger RNA are repeated in different chromosomes. Okay, so when when a gene is repeated, if the gene is 3,000 base long, you got 3,000 of these repeats, consecutive repeat pattern. And this is not what I'm interested. Okay, I'm not interested in the protein. I'm not interested in this very, very long repeated pattern. So I have to eliminate all these things and look for only isolated 20 base pair patterns. Okay, so once you look through the file and delete all the long consecutive patterns, and I think and these isolated but clustered 20 20 base pairs, and these are what I'm trying to look for, and hopefully that the pairs, all the pair addresses are identified in the long non-coding RNA data file, and so this way you get all the pairs in an entire RNA data, and then I can because this long non-coding RNA. They are just a collection of different RNAs. When I'm doing search, I really don't want to do the long non-coding RNA individually. Okay, it's just too much work. So I just combine as many long non-coding DNA into a single stretch, and then I just analyze 4 million of them at once. And once I identify all the pairs, I would reinsert the annotation that indicates where the individual long non-coding RNA starts. And if I reinsert this back, and you can see, okay, here is a long non-coding RNA, and from the beginning or the end, how many of the different pairs are in it. And this one you can see the various pairs that's being identified. And what I would like to see that the entire long non-coding RNA would just be one pair after the other and connecting to a cluster. And so in this way, necklaces can be easily identified in the per address list. Okay, in the nematode, the data file is only 1.8 million bytes, so I can just analyze in one chunk. And the nematode has 3,000 linear long non-coding RNAs. And the search time is, I think, 5 minutes, less than 10 minutes. And I was able to identify 2,000 pairs. And then I want to go back and match to see how many of these 2,700 pairs matches with the list that I have. I think I found 107. Okay, so that's a nematode, and that's human. So the human, the non-coding RNA data file, which is much bigger, 77 megabytes, and with 57 long non-coding RNA identified. And the search time, I searched them, divide them in 4 million byte chunks, and it took about 2 hours to finish that. And then I was able to identify 33,000 pairs. But I use this one and compare with the microRNA list, got exactly 107 matches. That's bogus. Okay, I was trying to find out what was wrong, and so ignore this one. Okay, I have to work on it. Of course, I was hoping that if I can identify the pairs, then the pairs must have many of them must appear as microRNA. But yeah, this morning I was looking at this file and found that it do not match that well because pairs are 20 base pair patterns, and the microRNA they are 18 to 24 byte long. So just compare them as they are. They will not match because the length are different. So I have to fix the microRNA and chop them into 20 base patterns and then make the compare. So that's some work that I'll do later. I was hoping that I can do a better search this morning, but not enough time. In my cell computer model, pairs and microRNAs are instructions, okay? And protein coding genes are primitive instructions which produce messenger RNA. And necklaces are high-level instructions with lists of pairs. So this, I think, theoretically, as a computer, the computer would work this way, okay? Because forth work this way, okay? List, calling list, calling list. That works in forth. There's no reason why it cannot work in a cell computer. But it's just that we need to identify all these pairs or microRNAs correctly. Okay, let me just show you some results, and then call it morning. Okay, any questions? Yeah.
How did you choose 6 base pair 220? And it seems like it could be arbitrary for different
Yes.
things.
This is what I call the exhaustive search.
Okay.
I search all.
Right.
Okay.
Right.
Yeah. So you go to a data file. You take the first 20 base pair and compare with everything. And then you do the next one and compare everything. And so you go through the whole thing. So this is exhaustive, and I'm comparing all 20-bit patterns.
The fourth can switch between binary hex and all that.
Mm-hmm.
So the programs could be part of the DNA program could be written in base pair 10 or base pair 12. Or base
Yeah. That's no problem. That's no problem. Mm-hmm. Yeah. Mm-hmm. Okay, let me just try to show you the nematode. Okay, yeah. Here is the search pad, the match that I got from the nematode. Okay, and this is the when you search it, see the first 6 base are the ones that the first all these patterns are on the same thread. Okay, so you just identify everyone has the same they are in the same thread, so they have the same 6 base pair head, and you have to just match the rest of the 14 base pair. And if they match, and you can identify them. So here you can see lots of it, right? So these are you search down to the TT, right? So these are all the unique but repeated patterns. Okay, and they are spaced. They are they are not consecutive ones. All the consecutive ones have been deleted. Okay, here.
Dr. Jin,
Yes.
so I noticed there's a couple on there that are perfect matches.
Yes.
What does that tell you?
These are the. Let me see. These are the repeated patterns. Okay, so you expect it. Yeah, right.
So how many full match patterns did you get?
Oh, well, all these, I got 456,000.
Well, I was talking about the entire string. I see a couple of strings. Like if you go up to 078.
078.
See how those two are absolutely identical. The
Yes.
entire string.
Yes. They are. These are what I'm looking for. I'm looking for the these complete repeat and complete matches. Okay, and they are on this list. There are many more, so they are at different locations. Okay.
How many did you get that were perfect matches, like the whole string match?
Well, yeah, this is search the search the these are the sorted. Okay, so that these two has been identified, but there should be three. Okay, should be three of them, but only two in the report. I only report two. N-1. Okay, so the last one didn't get reported. But in many of them are unique. Many of them are unique. Many of them are two or three. But the unique one has another one, which is not reported. Okay, so all the one, all the single, if it appears only once, that's because it has another one. But the last one didn't get reported. But if you have two, that means there are three matches. Okay. Okay. Now, if you find all these match patterns, and then line up according to the address, okay, and this form this list, okay. So you can, and then the C column is a distance from this one to the next one. And this shows that these patterns are clusters. They are not too far away. They are not too close. Okay. And this is what I call necklaces. Okay. So you just have consecutive patterns, which forms. I mean, they are pairs forming clusters, and this is the necklaces. So here, for example, here 44, 31, 132, 192, 198. These are kind of clusters. Even they spaced out 323. I think they're still in the same necklace. When it spaced out to 1,000, well, that's probably another necklace. Okay. So this is the results that I generally have. Oh. See, and these are, big numbers, small numbers, interspaced. So, it looks like this is a necklace, and there's a big gap, and another one, and another big gap. And so I think this is how genomes should be programmed. Okay. All right. Thank you very much. Any more questions?
When do we see the release of color genes?
No color gene. I hate colorful. It should be just black and white. Okay. Yeah.
For those 20 base pairs,
Yes.
you only see this two. I think that if they're long distance,
Ah.
maybe you can reduce your data base.
Yeah. I think and you probably should look at the sense and anti-sense. Yeah. But but when the long non-coding RNA, these are expressed, so there's no problem of the sense. Okay, even they exist in different sense on the DNA, they would end up the same. Okay, so so I think you don't have to reverse the pattern and do the reverse match.
Another question is about you got, I think it's 30,000 pairs, right?
Yeah.
So that means in all your long linears, you got 30,000 yeast.
Yeah, right.
So is that match to hypothesis? Because I think it's too big. It is too big.
Oh, I think it is probably not too big. Okay, you see the list we have for microRNA for all the different species. It's 58,000. Okay, and I think the experiment-wise, it's very difficult to get the microRNA. So I think the data, microRNA data is incomplete, or we only catch a very small portion of the microRNA, and that's why I couldn't get a good match last year.
I think if your microRNA if you have a microRNA can match to set many non-coding RNAs, maybe it has some functional. That means microRNA have it.
Yeah. I think the current theory is that one microRNA may affect lots of messenger RNA. But my thinking is these matches has to be exact. Okay, there's no they call the fuzzy match. The microRNA, they have to match first 6 or first 10 has to be matched perfectly, and the rest of them can be a little bit fuzzy. But from a computer science point of view, fuzzy match is not allowed. Okay, the computer, when you write a program, there's no fuzziness. If one bit is wrong, the thing will crash. Okay.
CRC, error correction, redemption.
Yeah.
Bearing that in mind, we can continue this discussion over lunch.
Yes.
Let's go to the door.
Yes. Okay. Thank you very much. Yeah.
Thank you so much, Ding.
You're welcome. Thank you for giving me the time.